Before starting any program, it's a culture to seek the blessings of the Almighty. I request the students of Department of Biosciences to invoke God's blessing on this program.
दयमाडे ताये वाग्देवी दयदिंद नीयन्न नोडे वाग्देवी दयमाडे ताये वाग्देवी हितदि सन्मतिय श्रीमती देवी नीडे हितदि सन्मतिय श्रीमती देवी नीडे प्रतति जनेत्री भारती निदय माडे प्रतति जनेत्री भारती निदय माडे दय माडे ताये वाग्देवी दयदिंद नीयन्न नोडे वाग्देवी.
Now, Dr. M.S. Mustaq, Chairman, Department of Applied Zoology, Mangalore University, will welcome the gathering.
Very good afternoon to everyone who has gathered here. Our esteemed chief guest, Dr. J.D. Gatkar, Professor and Director, Paul Herbert, Center for DNA Barcoding and Biodiversity Studies. Our IQAC Director, Professor Manjunatha Patabi, and IQAC Deputy Director, Professor Vishalakshi, Professor Monica Sadananda, and Botany Chairman, and Chairpersons, Coordinators, Faculty, and students of various departments, Zoology, Botany, Bioscience, Biotechnology, Microbiology, Environmental Science, Food Science, and Nutrition and Biochemistry. I extend very warm and cordial welcome to one and all present here. I am greatly honored to address distinguished gathering of the skill-based workshop on participating in the planetary biodiversity mission organized by IQAC Mangalore University. On the behalf of Mangalore University, it is my proud privilege to welcome our guest speaker, Dr. J.D. Gatkar, Professor and Director, Paul Herbert Center for DNA Barcoding and Biodiversity Studies, Aurangabad. Sir, your august presence empowers our student and faculty present here to carry out research in frontline and emerging areas of DNA barcoding and biodiversity. I very welcome you, sir. It is my pleasure to welcome our IQAC Director, Professor Manjunatha Patabi, Deputy Director, Professor Vishalakshi, and Professor Monica Sadananda. Welcome all, sir. Welcome. I welcome all the participants, those who have come from different department, of chairman of department, faculty members, research scholars, students and non-teaching staff, media friends and invitees. I welcome you all. Thank you so much. We look forward your active participation in today's workshop. Thank you one and all. Thank you.
Thank you, sir. We extend our welcome to you too. Now, Professor Krishna Kumar G, Department of Applied Botany, Mangalore University, will introduce the speaker to the gathering.
ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. Good afternoon to all of you. My duty is to introduce our today's chief guest, Dr. Kedkar. Dr. Kedkar is a molecular geneticist who is a scientific director of Paul Herbert Center for DNA Barcoding and Biodiversity Studies at Baba Saheb Ambedkar Marathwada University, Aurangabad. He has established a center in 2010 with an aim to map biodiversity of India using DNA molecules as a key factor for differentiation. This is otherwise the DNA barcoding in simple terms. The scientific movement for revolutionizing the taxonomy towards rapid mapping of biodiversity was initiated on a global scale, popularly known as barcode of life. Among the few scientists who took this gigantic work, Dr. Kedkar was one among them during 2005-2006 and represented India as a member scientific steering committee at Canada. Additionally, Dr. Kedkar is a director for Center for Coastal and Marine Biodiversity, a satellite center of the university on the west coast of India. During COVID-19 pandemics, Dr. Kedkar established the biggest COVID-19 diagnostic facility in Maharashtra and contributed patient sample testing for over 5 lakh swabs. Dr. Kedkar is basically a biologist and obtained his PhD degree from Stirling University and successfully completed postdoctoral studies at Hebrew University of Jerusalem, Israel in 2006, Stirling University, Scotland 2009, University of Hawaii 2013, and UNITAR Hiroshima in Japan. He has visited over 40 countries towards scientific deliberations. He has been awarded with the prestigious scientific awards which include Marie Curie Fellowship, Boy Scout Fellowship, Crest Fellowship, UGC Research Award, Commonwealth Academic Fellowship, DBT Young Scientist Award, University Ideal Teacher Award. He has published over 107 high-quality research papers in 2 books. He is leading a team of 33 researchers in his institute and working in various areas which are of public significance. His work is mainly on the evolutionary biology of aquatic organisms. He uses a broad range of techniques like allozymes, microsatellites, DNA sequencing, and now genome-wide comparisons and modeling. He is working on about 500 taxa on the various molecular aspects. He also has devoted substantial effort in developing protocols to authenticate food using molecular markers as well as wildlife forensics. Other areas of his group is exploring whole genomic sequence data analysis of native species having commercial importance, then cancer genome and certain human diseases. I understand that he has volunteered to come here and interact with the faculty and students. That speaks of his commitment to science and concern to our students. I am sure the interaction will result in some collaborative work which will benefit both of us and the country at large. Sir, we are sitting in a place saddled between Western Ghats and the West Coast, and this itself is a grassland ecosystem which is very rich. Western Ghats is one of the biodiversity hotspots, and ocean diversity I need not tell you. So we are sitting in a very important place at the foothills of the Western Ghats, which is one of the biodiversity hotspots, as I already told you, and that speaks a lot about his presence here. With this, I, sir, once again welcome you to our university. Thank you, one and all.
Thank you, sir. The time that we had been eagerly waiting for has arrived. We request today's speaker, Dr. G.D. Khetkar, to take over the session.
Yeah. Good afternoon. Are you a little loud? Good afternoon again. Yeah. So, respected dais, all the chairmen from different departments, professors, faculty members, research scholars, and students. I take this opportunity to thank Professor Monica Saranandan for, in a very short notice, organize this talk. 3 days back, I was just talking with her, and with a very short notice, she could able to manage this program and provided this platform to me. In biodiversity, if you talk, we are having huge biodiversity, still estimates also not complete. And we are having plan to barcode each and every species. And, in that initiative, I want to involve this university. Some echo. So we want to involve this university in this initiative, not only university, every sector of this university should get involved in this initiative. That is the reason I am standing in front of you. In real sense, if we talk about the biodiversity or talk about the taxonomic science, our country don't have any structured mechanism to collect the information. It is, ground reality. There are several institutes working in same field. We are having over 1,000 universities like this, over 250 research institutes, many agriculture universities, agricultural institutes, as well as your Zoological Survey of India, Botanical Survey of India, then microbial like things. In Chandigarh there is one institute. We are having Animal Genomic Resource Bureau, Plant Genomic Bureau, then Fisheries Genomic Research Bureau. We are having Zoological Zoological Survey of India, Fisheries Survey of India. There are many institutes parallelly working, but all the quantum of work is not coming to one place, and that's why estimates of biodiversity really no one knows what is happening in. Another thing, if you look at the global initiatives, what is happening across the globe, scenarios are so different, and that's why somewhere we need to start about this. And this is a beginning. Through this lecture, I want to have the beginning for mapping the diversity of our country. If we look at the lineage time also, several time, researchers as well as senior faculties also face a problem. Like whenever I'm trying to identify or taxonomically classify any species, I'm having several doubts in mind, and I always feel whether I'm correct or wrong. But if you look at the lineage also, for classifying human being or human species, he took almost 20 years time, because in first Systema Nature, he mentioned 4, like a lineages of human being, and he was keep on evolving it, keep on revising it, and in 10th volume he could able to change very little, like white Asians, the white like Europeans, he could able to change just white. In that way, he could able to do in 20 years time. That means you see, even he was having lot of revisions and lot of mistakes been incorporated. Even he been blamed because he is classifying human being based on the skin color, and sometimes he been talk as a racist also, because his method of classification not been accepted by many contemporary workers. So controversy is always stick up with the taxonomic science. Whenever we are doing some taxonomic science, animal is not bothering what name you are giving or what you are doing with that, what classification system you are applying. But always it is true in taxonomic science, this scenario is always there. And we need to go with some agreement sometime, whether you accept it or not. But with the experts or seniors or peers, whatever, or the authorities, we need to get tuned with them. And that created several, misidentification. One species having several names, several species having one name. This is very commonly happening. Then the taxonomic science, which was developed like almost 300 years back by Linnaeus, still that system we are using presently. I don't believe there is any scientific stream which is following such type of system which has not been evolved over a period of 300 years. And that is one of the biggest curve, or one of the biggest difficulty in proceeding or in accelerating the discoveries related to new species identification and mapping the diversity. This is one of the important thing we are missing. Every branch of science using computers, every branch of science using modern tools, but taxonomic science still we were following the linear system, whether it is parallelly in plant system, plant classification, microbial, whatever we are using, the system is like that. One of the biggest another issue associated with this system is this is not a universal thing. Because if I explain, sitting in this class, if I ask every one of you explain how it is looking like, I don't feel either two people will have similar explanation for this or description for this remote. Because these are more person specific, and that's why specific, whatever linear system we are following, it is more related to explaining the characters or differentiating the characters. And that's why during 2006 first barcode of life program was launched. We were only almost 12 countries were contributing to barcode of life consortia that have been built first time ever in the history, and where we were using some molecule which is universal type called DNA. So for mapping diversity, differentiating species or discriminating species, DNA have been used, and that become a basis for developing the consortia for barcode of life. And these consortia initially were very few countries were partner for that, and now if you look at 2015 meeting, over 200 countries joined this initiative. So 12 countries in 2005-06 and over 200 countries in 2015 demonstrates the utility of DNA barcoding and robustness of DNA barcoding technology in advancing the taxonomic science. But if you look at the progress going across the world, we are lagging behind like anything. And in that context, I'm here to have some ideas, here to share some few of the views, how research is progressing across the globe and where we need to chase them and where we need to join with the biggest initiative so that we can have literally barcode for every species in India. It is well known, DNA barcode might have, everyone who is studying in life sciences might have come across this terminology, which is really widely heralded and having tremendous applications in advancing the taxonomic science. Here, small segment of DNA we are terming, it is applicable for microbes, for fungi, for like mammal, like our vertebrate species, invertebrates, everywhere, including plants. Everywhere that segment is standardized and pretty nice protocols have been developed and people are following it. So if you look at the global developments, whatever happening, most of the places we are using like remote sense like technologies to map the diversity or to have a global observatory system. In global observatory system, in many way, remote sensing like technologies are giving better insights for making the observatory system for entire globe. But in this observatory system, one biggest thing we need to put in is a life. That means the life forms, different life forms. And if we could have mapping properly, then this system may become more robust. Like using this system, we are monitoring the algal blooms, we are monitoring, we are having like earth events, we are monitoring forest loss, we are monitoring very accurately as well as phenologies. That can possible with time-lapse method, we can do it very effectively. Only one segment we need to make it more convenient to observe in this observatory system is a life. And for that purpose, global efforts we need to take into account. If you look at the observatory systems, whenever you want to see something or correlate what is what, earlier days only describing species was enough, but now we are thinking about interaction between species are more important, because just identifying species will not pave the way for their conservation, but we need to understand their interactions. For example, you can see in oceanic observatory networks, you are seeing two colors here. So yellow colors usually you need to be there to create that information, and this blue one you can observe from the remote sensing technology. So here is a part, all the yellow blocks what we are seeing in this figure, that is the part of a biological scientist or biologist we need to pave the way at presently. For few years, we need to work voraciously in mapping this, and then we will have full-fledged Earth or planetary observatory system. So for planetary observatory system, what we need to do, because we need to work so keenly. If we divide the landmass on this planet into tiny, squares of almost 150,000 square kilometers, so 1,200 squares we can divide our planet in that. And by dividing that, it will become a much easier task to map and even to have an observatory system more effectively. Not only that, if you look at the India, we are having almost 35 plus, blocks of 150,000 square kilometers. So almost 35 to 38 blocks we are getting. And for Karnataka, if you calculate, that is not more than 1.5 block, which is just really equal to entire country, Germany. So that much part is very much important to have something to do. And if we divide it in, like I mentioned, 1.5 block of 150,000 square kilometers, how many researchers will be required? And if we do this data, how effectively we can use this data that I am going to demonstrate. But selecting this place for me is very important, because as Sir was telling, this is a hardcore of Western Ghat and most diversity-rich hotspot of our country. And that's why we want to have some centers in this place, so that we can have really introduce into the biodiversity hotspot area, and a lot of species we can describe in coming days. So why we want to have such observatory system, and we need to have some coordinates on this planet. There was one story. This emerald ash borer was found in some Chinese region, upper Chinese region, in 1995. That was the only place it was observed. But what happened after 20 years? Another survey been made, and it found that its range been expanded to some part of Europe, and then Canada and American regions. Largely it has dominated, and that was creating a lot of losses to agriculture production. So single species can dominate and can snatch your food away, just having expansion in their range. If you are having better observatory networks and better documentation of such organisms, definitely it will be better. How we can decide the strategy will be much easier for humanity. And in that sense, such observatory networks are very much essential for the welfare of the humanity. If we create such type of database, to be very honest and very straightforward with you, we don't have baseline data about the biodiversity in our country. We need to set somewhere a baseline. Without baseline, we can't say that what phenological changes happen, or what range expanded or range shrinked for a particular species. Whether it is, you take example of anything, whether it is bacteria, fungi, any disease, or you can take even mammals. So for that, we need to decide on something which is foolproof, and it should be documented, and globally it can be linked. And for that purpose, there is a nice powerful tool is available at our hand, is a DNA barcode that can have many opportunities to link your species interactions, comparisons between regions, comparison between states, between countries, and among the globes. It is pretty possible. And that's why if we are seeing here, like I'm mentioning 2020 and 2050, I can give any cycle, year-wise I can divide data, 10 years, 5 years batch I can give, or 50 years, or even 100 years, lapse time we can map, and you can have one day such type of data available. Coming generation can harbor this for years to come. And then it will become more effective for policymakers or governments to decide a conservation policy. Presently, we don't have solid data available with us to decide a conservation policy, or to decide some efforts, or to recruit some efforts in conserving particular species, or reducing the helm of a particular species. And for that purpose, we need to decide some baseline. And this is not like a very small job. Another important thing, we must look at this, and we must understand, biologists had never taken immense efforts. Like if you look at the astronomical sciences, they are having pretty much a good number of projects having billions of dollars invested in astrological expeditions or astrological research. If you look at the physics also, there are many multi-billion projects they recruited, where a lot of investment happen and they try to advance something. In space also pretty much investments happen. And in biology, there was just a single project which is having a quantum of more than 1 billion. No another initiative or global efforts been taken in biological sciences to do something on a vast scale. If you look at the top three expeditions or top three research efforts, they are looking at something which is not existing or something which is far away from us. We want to, some biologists want to create something, to see something in your backyard, that we are not taking efforts, but we are seeing something on the Mars or something in the moon, but we are neglected to see something which is very much important for you in your fields or in your environment. What is there that we are lacking? And there is a failure of biologists, where we need to think seriously to have some initiative where we can do some massive efforts. If another example related to this, earlier days, like even our teachers were telling, more brain, less instrumentation, good research. It was the equation our teachers were telling. But now if you look at the developments, current developments for last 2 decades, if you are having sophisticated instrumentations, data generation will be more intense. This is a formula nowadays people talking about, and this been mentioned by George Hale. Without sophistication, you can't create a usable data or reliable data, and for that purpose high-end instrumentation is very much essential. We are talking about a diversity of a mega diverse country like India, having several biodiversity hotspots, many seasons. And if you go from east to west, or north to south, if you go many climatic zones we are having. And in that sense, if you want to do something like a traditional way, it is very impossible to do the biodiversity mapping. If you look at some big discoveries, what happened in case of this Large Hadron Collider, 10,000 researchers joined together, and they were finding what little charge on electron. And because of this investment in the collider, Hadron Collider, they could able to advance energy up to over 1K. They could able to do it in a very short period. Similarly, your Hubble telescope, you must be knowing, many researchers, over 2,000 researchers joined this initiative. Earlier days, this was the scenario resolution of our galaxy. Now, how much resolution, 1,000 times or even 1,000,000 times resolution we could able to do. This happened because of technological interventions, or this happened because of hyphenation of the technology and joining many minds together. But in biological sciences, we used to remain in close premises. We don't interact with others, and we are having very poor collaborations to explore something on a broad scale. And this way we usually used to restrict ourselves in doing taxonomic research or biological research. Our requirements are like very few. We need some key books having the taxonomic keys. We are having some type of magnifying glasses, and then a small microscopic devices, and using this we want to do something which is as comparable to other big discoveries. We want to do something with these mere facilities. Really, such things are really not enough to map a huge biodiversity. To have more insights on biodiversity, we need something like an industrial scale. And in that way, DNA barcode, which is the first technology where we explore DNA sequencing. For that, now we are having very robust technologies and instrumentation available like NGS facilities. So there are many platforms available where we can do industrial scale explorations and map the diversity in a very short period. If you look at the developments in the taxonomics, like biodiversity science, so over 30,000 vertebrates, means from 70s we lost 60% of the vertebrate species. It's a big number. And if you look at the insect, this story is regarding the Germany. Many insect species they don't found which were there 5 years back or 10 years back. Almost these species were vanished. Why this vanished? There is no answer for that. Similarly, if you look at the Argentina, they lose almost 2.9 million birds. 2.9 million is a big number, and this much bird species vanished. No scientist can have ability to bring back any species which is extincted. And in that sense, if you look at whatever development happening in the biodiversity science or taxonomic science or biological sciences, we need something which is an industrial scale. And without that, we will have several species with us, but we don't know they are here or where they have vanished. Without our notice, many species may go extinct. And in that, that lieu, the International Barcode of Life Consortia launched, and the mission was International Barcoding Consortia mission was to have Earth observatory system, to create Earth observatory system for better planet to next generations or coming generations. In this observatory system, what we were doing in this consortia, plan was 2020 to 2045. It was like to register every species, document their interactions, and monitor their populations. These are the 3 aims decided for this. And in this 3 aims, first was the barcode, 500,000 species, and that been successfully been done. India contributed greatly in that, but very few institute, become partner for that. And another program was bioscan. Still we are the member for this. And third program we want to launch from 2026 to 2045, where literally every species should be mapped. I was showing you initial things. Even there are many mistakes happen in the species discovery. Mistakes happening is not a big issue related to registering a species or registering the biological form, because Barcode of Life Consortia has so solidly been built on bioinformatic platforms, where whatever sequences you will deposit, they will specifically classify based on the bioinformatic knowledge, and if it is identified somewhere else in the world, so it can be mapped with that name. If it is not mapped, it is completely new species to the world, then it will be assigned to a bin system. So barcode index system is there, BIN system. So barcode index number system is already implemented, where if species is not identified, just you will have that bin number. Whenever in time and space, whenever some researcher will identify that species, the unknown species will have its name. So it doesn't mean like if I am today depositing something, I am doing some mistakes, or I should not deposit without name. This biggest trouble been solved by the bin system in the barcode of life data systems. Then by doing 500,000 species discovery, India is contributing almost 30,000 species, and in that 30,000 species alone, Aurangabad Center is having contribution more than 22,000 species mapped. So over 90% data is deposited alone by Paul Hebert Center in Aurangabad, and remaining, some 10-12% data is deposited by allied institutes entire country. But by doing this, like 5 million specimens been collected globally, which belong to 500,000 species. And by doing that, now lot of applications we are discovering for DNA barcode. Even now, most of the publications also, reviewers asking for DNA barcode to be submitted along with a species, taxonomic identification. That much application side this is having. Applications, if you look at many places, DNA barcode is now very, fluently used in used in your forensics for food or wildlife forensics. Then it is used in disease diagnosis, it is used in species interactions, discovery of new species, finding cryptic species. So many applications we are having, and several papers coming every year, several papers coming. One important thing about the consortium of Barcode of Life, 200 countries I said joined this initiative, and over 20,000 researchers joined, and how much funding we are expecting for that entire initiative is not even near to billion. So even small efforts also can reflected into good outcome. And publication wise, if you compare DNA barcode initiative, that is even more comparable than your Hubble telescope or your Hadron Collider. It is more than that. Citation wise, if you compare, that is also true. Citations also, we are having more citations in barcode research papers. Important thing, earlier research, whatever we are talking about physics or we are talking about the space science, most of the rich countries were contributing to that. But beauty of DNA barcode is even countries like like Africa, they are also contributing equally to the developed countries. That means this is a widely acceptable technology and widely trusted technology by the biologists. If you look at the barcode, whatever 2015 we were having the first assessment about the species, whatever been done, you can see every species, every dot is showing a species. Red dot means over 100 species at one place. So it been mapped very nicely. Why we could able to map this on a global region? Even a particular 1 square kilometer, 1 square meter area also can be mapped to one species. It is possible on this map because we are collecting very nice information about the species which to be registered with the DNA barcode along with its coordinates, and that's why species occurrence can be pinpointed very accurately. If you look at the how many specimens we are having, like over a period of time, that has increased. That means a lot of people involved in it. And very nice, bioinformatic platform. Like now our life is, driven by the search engine. You agree or disagree, but life is driven by search engines. So first search engine we launched for taxonomy is a barcode of life data systems. That is a taxonomy browser or taxonomy search engine where you can put species name, where you can put a species sequence, and even image wise searching also possible in this search engine, so robust. And using this information you can create like a barcode index library for your specimen if it is totally new. If it is already identified, then it will go to this species level or whatever taxonomic level it will get identification. So automated species identification become pretty possible by very small technology intervention, and this is doing very effectively. Now International Barcode of Life Consortium initially started working from 2016 to 19. It was like a bioscan program. India was there. That map is, I have not included that map, but India was the part of that. And Planetary Biodiversity Mission, we want to have initiation from 2026. And for Planetary Biodiversity Mission, what is mean by Planetary Biodiversity Mission and what we want to do? Like I told you, in some cases, organisms DNA barcode based identification approach was using Sanger sequence based data, where we are taking single organism, observing it, taxonomically describing it, taking little tissue, and then taking DNA, sequencing individual specimen on a sequencer and then putting that data. That was pretty laborious and much expensive task. Like if you see here, up to 600 rupees per species required for barcoding a particular species, and every individual you need to do. Now there is a new technology in the form of a next generation sequencing came. And in this next generation sequencing, in Sanger I need to have only one specimen at a time, one PCR, one gene. But in meta barcoding or meta genomics like technologies, what we are having in next generation sequencing here, 1,000 species DNA you can combine together and run at a time. The cost of sequencing would be very much less. Even I am showing here is a pretty high. If you are having more number of samples, cost can go up to even 60 rupees per specimen or per species. That is possible because of meta barcoding. What we are doing in meta barcoding, we are taking all species together, taking the parts, isolating DNA, putting different markers, amplifying it and putting into sequencer and getting all the data. And that data can be directly classified using bioinformatic intelligent tools and can be mapped. And whatever background you are having about your species can be mapped perfectly on the geographic coordinates. So this is the beauty of having next generation sequencing technologies. There are 4 platforms presently available in next generation sequencing technology. One is a SQL, little bit costly affair. Another is like a Illumina short read, and this is a wrong lead. In case of SQL system, you are getting sequence up to 50,000 base pairs. Although its error rate is very high, but in case of MySeq and NovaSeq, you can do up to 600 base pair sequencing. Error rate is pretty less, about negligible, and this can give up to 600 base pairs, where you can have millions of, like 100 million species you can run in a one program. So one run 100 million species, just imagine how many species are there in our planet, or talk about India. So pretty nicely we could able to do planetary biodiversity mission is really possible by having these industrial applications of next generation sequencing technology. So in next generation sequencing technology, most of the cost is going with its flow cell, and then rest of the things we are doing like a traditional way. You need not put any expertise is also not much required in this because they are having tailor-made protocols. You just need to follow the platform, the guidelines, and you can reach up to the sequencing. Just this bringing machine and having some consumables or its flow cell is a just little costly affair. But when you are doing some million species, that I think this cost is negligible. Then by doing this, you can have up to 24x in 384 well plate. You can put every processing you can do in this way. Specimen, you can take specimen, extract DNA, then PCR it, put like small tags, amplicon, then pull amplicons together up to 9 to 16 pulls you can combine together, and you can get a barcode. Even whatever format you required about your species, that nicely you can resolve over, and base data can be generated. Now planetary biodiversity mission, basically what I am aiming at, we want to select in our country, there are over 1,000 universities I mentioned. So initially we want to select 120 universities entire country, and from these 120 universities we will select 6 nodal centers where NGS facilities will be installed. There will be one intelligent portal we will be designing now. In that we will register our interested researchers with their biodata, everything, and we will develop one national portal for India. And one national program will be initiated where we are going to ask around 60 to 70 crore rupees. 6 facilities will be there where all these facilities will be installed, and whoever researchers come with their samples will have free access to the sequencing facilities. And for this, all bioinformatic backup will be provided at one center. All databases, generation, data repositing at one portal and linking to biodiversity planetary mission will be another aspect of this initiative. So that way, structurally, we want to go for mapping the species where what expertise you are carrying, you find your collaborators, bring your programs. Majority of the funds we want to create for fellowship for the students, salaries, travels, and consumables, in addition to the basic facility. This is the aim of India Planetary Biodiversity Mission, and that we want to complete in coming next 2 years. Before 2 years, we will have every facility ready, and we will launch our program from before 2026. And we want to aggressively participate in the mapping the diversity of our country, or barcoding every species of our country. Whether it is known or unknown doesn't carry much meaning, but we want to register what is there. And in successive period, whoever researchers interested will try to advance the knowledge what been generated by Planetary Biodiversity Mission India. So for this purpose, we want to have such type of system to be created. Now, if you see in Planetary Biodiversity Mission, I was telling you 1,300 blocks we are having. So every block, if we map like this, and samples collected from that block, and if it is linked to sequencing technology and data being taken out and deposited to the repository, how beautiful things we will have at hand about our biodiversity. You can do many comparisons. You can have multiple applications. Practically, theoretically, you can do things, and you can bring out newer knowledge about the biodiversity of our country. So by creating 1,200 sites globally, I mentioned in India just 38 sites we need to create. By creating this in 52 weeks, theoretically it is possible to map every species on this planet, entire world. It is possible. So why we don't think that 38 coordinates we can map in coming to 4 or 5 years? I think it is not something like impossible for us. And that way, if we could do, so like cost per sample, they mentioning 50, cost per sample I am mentioning 50 euro. That means in one sample you will have sometime 1 million species. So what one species will have cost calculated? I think this is pretty negligible cost, and any institute in our country or any researcher can afford it. Possible. And by doing this bulk sample per year, like it is possible to even for entire world, 62,500 samples you can collect. What will be quantum of India then? Maybe if you are having one sample per coordinate also, if you are going, our sample number is not reaching even 50. So we are mentioning to have 6 facilities. 6 facilities actually does not required, but even if our estimates if go wrong also and multifold specimens if we found also, in that case, this is rational to complete this task in a very short period and have illumination of biodiversity in our country. There are nice, bioinformatic platforms already available for meta barcoding or for, general barcode data. By doing whatever data you are submitting, you can have resolution about the species, within species differences. Then between conjures also you can find differences, several discoveries. When you are having conjure difference, is more than 3%, there are many cryptic species may be present. So you can rush immediately to the taxonomic expert. See, I am getting this much data. I suspect there may be many species present in this group. And that way, your discovery can have more vibrant way to map newer species with a solid support with a genomic data. Similarly, if you look at the Costa Rica, here also they are having like a between conjures distance is very high. It is always pinpointing your species identification is wrong. What name you provided may have many hidden names and you can resolve it. But like whenever we are creating sequence clusters using this data, you are not doing anything, you are collecting samples, putting to machines and then data is depositing. Whenever you are depositing data, you are creating sequence clusters. And that clusters can be then linked to the species if known. If not known, then they will go as a cluster. And that cluster, whenever someone will do work on that, that cluster will get name and your species will also automatically get name. This is called the integration of minds, integration of laboratories, integration of nations and integration of researchers. So all this is possible by doing nice informatics in DNA barcode. In DNA barcode, whatever data we are repositing now, we are putting many things like if we are having one species, you want to see where another part of the world such species found, immediately you can get such information. You can have species boundary, you can have like a barcode gap also to find out whether your identification is correct or not. All type of specimens, who collected them, who is a researcher, where it is deposited, where I can get the photograph or access to specimen or its tissue or DNA. That every information is at one click, and that way it will create a nice story. Not only that, if you want to compare one species found in Mangalore, you want to compare it with Maharashtra or Delhi or any state or globally, that is possible like this. How much there is a sharing about one group of species between countries, between continents, between states or regions or laboratory, everything comparison is possible by depositing your data to barcode consortia. Then most important thing I will tell you why species are wiped out or why species are going extinct, because when the species having interaction, if it is vanished, so definitely species will wipe out. For example, honeybees, you take an example, if you don't have flowering plants and not having much nectar, which honeybee like, so definitely honeybee will become orphan and one day that species will wipe out. That is called a species interaction. If we don't know species interaction, then how we can conserve it? And that's why in the Bioscan project and planetary biodiversity mission, one major objective is to species interaction. And by doing species interaction, you can see many things and then you can pinpoint what are the conservation measures you need to impose for restoring or conserving a particular species. So first we are having plans like species samples we want to collect from different regions. Then species interaction we want to do with 5 million specimens. Then we will have species dynamics based on the ecoregions where almost 100,000 bulk samples need to be collected, what 53 I mentioned in earlier slide. And that way we can have scanning the entire planet. So many places we are having, like if we mention like 8 million projected, earlier days it was 7 million species on this planet, then it became 8 and now we are talking many million specimens are present on this planet. And how many at risk you can find out based on the your ecoregions. So that way we can predict out what are the species, what quantum of species are at risk globally. And in that sense, if you look at today's estimates, it is saying that there are 18.1 million taxa which is dark. Dark means we don't know. They are there, they were there, they are there, or they may be there, but we don't know. We don't have measures to map their existence. And what we know is very much, little we know about that. It is a tip of iceberg, what we discovered. Imagine, after Linnaeus, he started the system of classification or classifying organisms or knowing organisms, 300 years gone. In 300 years, we could able to do this much job. In this job also, 90% is share coming from DNA barcode data, and remaining is a morphological data mapping. So how much this technology has contributed and how much we need to contribute is 18.1 million taxa we need to concentrate on that. And for that we don't have anything we don't know. And by finding this taxa, we will find out more on the interactions between the species, and this will become a major tool in conservation and major tool in creating the planetary observatory mechanism for our Earth. So multicellular, many species, we don't know how many multicellular species are there, like plants 0.5 million, then fungi like 5 million, and animals 15 million. In this way, if you took and that how much interaction they are having, it's a big task. It may take millions of years to unwind. But if you look at the some of the nice classification or species discovery move been done during past 10 years. If you look at this Arctic region, 10⁴ species been discovered. Europe 10⁵, and Costa Rica, much smaller region, they are having 10⁶ species. How come this disparity has been found? Very interesting thing is, he is a Danjan from Costa Rica. He and his wife and few bunch of researchers, they could able to map entire species of Costa Rica. Although country is small, but they are having big resolution for entire country, and now already they started planetary biodiversity mission. For that they got 25 million from their federal government. So only one family, they were doing this research, entire country they mapped, and this is the first country in world who is having every species mapped using barcode like technology. And now they are dreaming to have species interactions, and they are having so many beautiful experiments they did. Like in Costa Rica, they did small experiment like this: cloud forest, dry forest, rain forest, moist forest. They took almost 4 to 5 grams of soil per week, and they tried to map seasonally, and they found tremendous data associated with that regions. So every eco-region is a specialist region. You can see here they did like species accumulation curves. For 4 eco-regions, they are having over 20,000 species they could able to map, and they go to different beans. That means still they are not given name. And they are having barcodes up to 200,000 barcodes they could able to do. Alone, one Danjan, he is a scientific leader of that expedition. And when they are having like barcodes and bins for different region, just distance between these two is 20 kilometers, 6 kilometers, and this is again around 20 kilometers. In this region, what much species accumulation you find is so much different. What our understanding about diversity is even smaller than a tip of a nail. What we are talking, we know everything about, like biodiversity of Western Ghat or biodiversity of something. I am working on biodiversity, or sometime we talk XYZ has explored entire biodiversity. If we look at this example, I don't believe we have even touched 0.00140. That much percent also we have not covered about the diversity. Forget about the interaction and forget about the discoveries about new species. So in that sense, it is important to see the species interaction. Why 2 species, 3 species remaining together, who is associating where, and how it is creating the symbiotic relationship or whatever type of relationship to sustain each other. This is very important. You must be knowing this is a popular story where these caterpillars have been caught and they were classified to one species. And taxonomists were claiming they present different habitat, and they are feeding on different plants. That's why they are having different coloration. When DNA barcode done, we found this 10 as a 10. Every individual is a new species, and that way one species turned to 10. So that in that sense, whatever interaction everyone is having is completely different, and that interaction, if you try to map using earlier barcode based technology, it is difficult, because Sanger based technology, when you are putting more than one DNA, so you are getting co-amplification and you can figure out, and usually we were telling this is a contamination of DNA and we are throwing it. That was like a most important information we are missing, but technology was insufficient. But when we are having this barcode next generation technology like SQL or Illumina, then such type of deep divergences we could able to map. And what is these deep divergences? You can see there is a co-amplification of the host gene. The elephant is a host for this mosquito, and when I am taking this mosquito for barcoding, I was co-amplifying some genes from this vertebrate host, and I was getting deep divergence, and I am assuming something is wrong. So this species interaction I can do very effectively using new technologies, and that is pretty possible. You can see here, this is from Africa, this is from like Australia, and many, like these are the nematodes which are associated with a particular host which is infected, and that divergence you can see up to 43%. So species interaction become powerful tool in mapping these species. Like you can see in this figure, we are having vertebrates, arthropod, nematode. For everything we are having standard set of genes, mean protocols finalized. You take plants, you can take fungi, you can take most of the other taxa or Olbochia, whatever you can take. For that we are having a specified standardized markers. Now you need not worry about selection of markers. Accurate marker selection and amplification is pretty possible, because we are having universal pair of primers for every group of taxa. Then protocol is also present there. If you take all this information together and if you sequence, your bioinformatic program can give better resolution, and it can tell about the species interaction more effectively. So this is possible, and this can become one of the powerful tool. Can you imagine? We are the host for over 229 parasites, and what interaction everyone is doing with you, and how both are evolving hand in hand, or co-evolution is happening. Well, very nice story you can create like this. This is just for one example I am showing you. Round worms, we are having like different round worms. We are terming them as a one parasite. In that sense, if you look at this, several interaction networks we can find out. And doing this, like you can have interactions, you can have like whatever symbionts available with that, and that how these symbionts are differing from their host, it can do very nicely. Like you can take Lepidoptera, you can take Nematoda, Diphthera, Hymenoptera, whatever. If you look at the developments, whatever happened across the globe, most of the research been explored, they added the data associated with the insect. Unfortunately, in our country, there are very few insect taxonomists, and that data been not barcoded. So if you look at entire barcode of life data, 70 to 78% data is coming from insect, and unfortunately that total is lacking in India. We need to create a robust system, and we need to map every species by involving several researchers, students, faculty members, and many institutes. This is very much important to do to map out such type of better things. Then what is the present situation? We are having like best industrial scale technology available. We are having softwares, and that been aligned with the barcode data. We are having like community, you and me are there, who are working in the field of biology, and in that term, what we need more than this, just we need to explore little funding opportunities, and by doing that we can build a nice planetary biodiversity community for India, and we can do this research in a record time. It's pretty possible. Earlier it was decided for planetary biodiversity mission. India was not decided, because what every planetary biodiversity nation should have able to send their samples or bring samples. India is not selected in this because we are having a very stringent biodiversity act. We can't send our samples outside, or we can take samples from outside in. And that's why India is not shown here. That's why we need to build our capabilities. If we don't build our capabilities, you can't send your sample outside, because biodiversity board will never give you permissions in that way. Or some another country also can't send samples to you. We need to solve our problems ourselves. And for that purpose, this move to map Indian biodiversity or planetary biodiversity mission India, we need to set very quickly, because other countries will, explore their possibilities and can have mapping the species interactions. They will have global observatory program, but India will be excluded from that satellite imaging. If we don't want to happen like this, we need to work together. We need to work from today, and we need to do something. I think it is affordable for us. 60 to 70 crore is not a big amount. Even we are having some discussions at the secretarial level in central government. And I got very positive signature, positive indications from them. We can launch such type of program. So in that way, I'm just having such meetings with most of the important universities where we want to set our centers. This is 7th meeting I am conducting. And through these meetings, we want to identify, we want to make people to think on this. And slowly, one by one step, we will launch our portal. We will have, connections between us. We will launch everything, and then we want to, first we'll complete the South India, then we'll spread over to the northern parts, northeastern parts. And by doing this, one day we want to launch this program, and a mega proposal will be submitted to Government of India for funding. Not much funding we are expecting, 60 to 70 crore rupees we are going to ask, with very nice structured proposal. And we will incorporate each and every aspect, every university, which university we will select, that will democratically we will do the way. We will put principal investigators, co-investigators, their fund requirements, their area of specialization, everything will be linked together, and then we will set another training programs across the country for research students, for master students, for postdocs, for faculty members. And then we will launch Planetary Biodiversity Mission for India. And hope once we will launch, within 5 years we can cover almost every species of our country. And that is like a towering goal we are setting, and for that purpose I am here. I think globally 600 million euros, 500 to 600, that means totally they are expecting 1100 millions. We are expecting just 60 to 70 crore rupees, which is not a big deal for government. It is like a peanut. And for that purpose, all the senior faculties, I request you, whatever your capacity, try to write some articles in newspapers, so that your government should listen it someday. Small news articles, if possible, write in your local newspapers, that should reach to government. We want to have planetary biodiversity mission. What is use of planetary biodiversity mission? Why we want to do this? So all these things should be covered in your articles. And try to find out your groups, how we can go ahead, send us suggestions. Very soon we will going to register you. Any number of students or faculty members we can register on this portal, and from that, based on biodata capabilities, we will shortlist people and incorporate them into the main proposal. That center, what 6 centers we are going to select, we will carry out the work for other rest of the places, rest of the institutes, individual researchers or NGOs. They will send samples to these centers, and data will be created, scrutinized, bioinformatically analyzed, and will be deposited to Planetary Biodiversity Mission India, which will finally be linked to the Global Biodiversity Mission. This is one of the ambition we want to do. By doing this, what we can do, we will have the identification of dark taxa, reporting of dark taxa. We can find out their interactions. We can have observatory for country, your state, your region, maybe Western Ghat Observatory, maybe for Karnataka Observatory, maybe agriculture diversity, whatever. So in that way, we want to create an observatory for entire country. And by doing that, every species will be named with your presence on this map. If you look at this map, here you can see the dark red mark. That is a contribution of our laboratory. Only I started without facility. I was not having single facility till 2010. After 2010, we started efforts in building the capability, and now my center's investment has reached almost 100 crore rupees. Single-handedly, we could able to do this. What I am showing you, the next generation sequencing technologies, they are Yeah. So what I am talking about, the next generation sequencing technologies, all these platforms are available at my center. We are providing services. We involved many people. We conducted several training programs. One training program was conducted in your city also in 2014, Mangalore College. And such 24 training programs throughout country we conducted. We inspired several young minds, lot of people, 70 collaborations we built so far, and lot of data been deposited. You can see this data deposition could happen because of our collaborators. Many collaborators working together, but quantum has not come. Reason, very few institute involved. Funding was an issue, and DNA barcode, like a single gene-based data analytics we were doing, which is time-consuming and expensive also. Now we want to explore with a bigger industrial scales with the NGS technology, and we will have 6 centers addition to Aurangabad center. And that way we want to illuminate the diversity of our country. I believe this is not impossible. Pretty possible we can do it. And for that I need your help. I need your involvement in this initiative to build our nation better, to make our nation better available to coming generations. With this, I thank everyone. I must acknowledge Paul Hebert, because most of the data resources I am using from his source, his presentation in this presentation. Many slides I use from his own presentations, because he is a consortia leader and he is the father of DNA barcoding, who lead the entire globe for DNA barcoding initiative. And we want to have better representation in global scenario. We don't want to lag behind in planetary biodiversity mission, and we want to illuminate our country in this entire map. So join together, we will do it. Thank you very much. Questions? Yeah.
Imagine a world in which you can identify any, on the spot, in an instant, anywhere on our planet. And this is the world that DNA barcoding will build. Thank you, sir. The session was so enlightening. You enhanced our knowledge on DNA barcoding, species interactions, technological interventions, speciations, beauty of next generation sequencing, bioinformatics tools, so on and so forth. Being life science students, I'm sure it is a wake-up call for all of us. Let's all join hands to contribute a part to the environment. At the same time, instead of focusing much on technological innovations, it's better to conserve which is already existing. And now the session is open for discussion.
Yes, sir.
Sir, thank you very much for your educative and inspiring talk. My question is, a couple of questions are there. GenBank sequence database is also there. Is there any link with the barcode consortium? That's one question. Second thing, traditional taxonomy is already there. What is the status of traditional taxonomy for this one, barcoding? How they can contribute? For example, we have plant taxonomists, animal taxonomists, or in a specific species specialized taxonomists. How one can contribute through barcoding consortium?
Yes.
And another thing, how many new species have been discovered based on DNA barcoding, global level or in Indian level, like that? I want.
Okay.
Thank you.
Sir, new species discoveries, that rate I was telling you, we are comparing like 300 years data, and along with that we are comparing the DNA contribution. So tremendously, 1/3 quantum is been discovered during this 10-15 years time. Biggest contribution been done. Then sometime what happened, I was assuming, for example, we are working on a catfish and Clarius. So we are not knowing, we are terming Clarius batrachus is a species available. Then in northeastern region we found some differences that become another species. Again we are having some more collections, we found there are many species present in one Clarius. It become a species complex. Over 6 species now we are assuming in that one taxa, which we are assuming as a single species for years together. So that is a beauty of species discovery using this tool, because here morphological taxonomy, you can quantify the data. Here you can quantify the data. Like I was mentioning here, you can have intraspecific distances. And there is one gold standard set, ki if your data is exceeding 3%, then there is another species. So that way we are having quantification in barcode data. So this way we can contribute greatly. And regarding the experts, if you are expert, that will be better then. We will have our sound reference library creation. If you are not expert, then that case also that data will go to bin system, and someone who is expert will explain someday, and your species will get a name based on this sequence match. Yeah.
The common genetic marker used for coding, other mitochondrial oxidative one, that is common used for DNA coding. Other than genetic
I am not understanding, sir.
code. So usually mitochondrial cytochrome oxidase one is DNA
Yes.
barcode. So other than this genetic marker.
Yeah, there are many markers available, sir. You can even sometime you can include the nuclear markers also along with that. If you are not having pretty sure about the differences you are getting, then you can use multiple markers. So in that's why SQL, why we were mentioning there up to 50,000 base pair data you can incorporate. So meta barcoding you can do using one gene, or even it is possible by doing entire genome also. So what is your objective? Based on that you can do. But in barcode of this planetary biodiversity mission, it is open for all. Means you either you do for entire genome, or you do for a single gene or multiple genes. It is open for that. Yes.
Hello, I am Dr. Mushtaq from Mangalore Department of Applied Zoology. Actually, DNA barcoding is used for only we can differentiate cryptic species. But when we want to go for paper or new species, all the halotype, everything we have to deposit.
Yes.
So there, DNA barcoding only is not the case. We need only even traditional.
Yeah. So this is a typical taxonomic question, taxonomist question. Actually, I agree with you. DNA is not a solution for everything. I know that. But in time and space, we need to discover our species which are vanishing rapidly. So at least registry will happen with this initiative. Then there is ample time to further rectify the issues related your halo species or cryptic species or new species or sister species, whatever. So for that we can do the discoveries, but at rapid space, when you want to create a national observatory, biodiversity observatory, we need to contribute in this way, so that at least you will know that this was found in this region. After 100 years, it is not now here. Then we can go back and see what changes, phenological changes happen to that region, so that this species is not finding here. So that way, beautiful applications you can do in conservation science.
One more thing, here in our country, very difficult to get the samples. Like, for example, we have threatened species, and is it easy to get now for your mission? For example, civet or tiger or anything, we have to get the sample. Okay, maybe we may get fecal samples, but other sample getting permission.
Yeah. So, sir, this planetary biodiversity mission, we will align completely with the government regulations, and that's why we want government funding. Even for running this show, we may have opted some foreign funding, possible. But we don't want to do that, because we want to align with our regulatory policies in India. And when project is from regulatory bodies, we are getting the fund. Ultimately, we will get inherited permissions to collect the samples for completing India planetary biodiversity mission.
Already our already identified. Their samples are there in our museum. So we
Yes.
can also add it for this planetary biodiversity.
Yeah, sir, this is a good question. Many places we are having old samples or preserved or curated samples. There are protocols which can get DNA out of that. So that also been Barcode of Life Consortia. They develop protocols for getting old samples or even these formalin preserved samples. Also, DNA obtaining DNA is possible. But question sometimes is coming like quality of your sequence remain poor. But we can take efforts in doing that. I think Zoological Survey of India, Kolkata, they are now taking one initiative to take back their samples from the repository and do DNA barcode. So similar line what you ask. So that way we can create. I will tell you though so much interesting things in these discoveries. We are working on gangetic dolphin, and in gangetic dolphin, getting samples from India is okay, but getting samples from Pakistan is not possible. There are two subspecies of gangetic dolphin. One is a Platanista gangetica gangetica and Platanista gangetica minor. So that sample getting was very difficult, but from NCBI I could be able to download the sequences, and then we could mention in one of our publication, by comparing the sequences, we can mention that there are no two subspecies, but that is a single species of Platanista gangetica. So this much flexibility, sequence data can give. For sequences, you need not require permissions. That you can obtain from the search engine easily by NCBI like GenBanks, and you can use that data. So likewise, people will use our data. People can comment on that. They can correct it. Sometimes suggestion can happen. Thousands of taxonomists now join with DNA barcode initiative. So you are not alone working on some specific species. Lot of people will join with you in that. Yes.
So nowadays the DNA metabarcoding is being slightly integrated into the field of forensic biology. So my question is there any limitations in the DNA metabarcoding?
Lot of echo I am getting.
Is there any difficulties in integrating
Can you come this side, please? I am getting lot of echo. Any difficulty in? No. My laboratory so far published I think seven publications on forensics. Even many cases we got a conviction. Means the data we analyzed for police cases got conviction in the court. There was no cross questioning about the authenticity of DNA barcode. It is foolproof, and all our papers are available online. You can check about the forensic. It is related to food forensic. It is related to wildlife forensic. It is related to rehabilitation of species. So all this data already we published, and it is available freely. You can download it. GD Khedkar you can mention, and DNA barcode, you will get our papers. Good question. So if questions over, very soon we will launch our portal. Join this portal. We'll let you know when it will be completed. Join this along with your specialty, your capabilities. In that sense, we will complete it, and then we will complete South India. Once it will be completed, that one portion, whether we will go region-wise to Government of India, or whether entire researchers will go as one entity to Government of India, we will decide in coming time. Thank you. Yeah.
Thank you, sir. Now, Professor Monica Sadananda, Deputy Director, IQAC, Mangalore University, will extend the vote of thanks.
dignitaries on the dais, it is indeed an honor to propose a vote of thanks today. On behalf of the IQAC Director, Professor Manjunath Pattabi, Deputy Director, Professor Vishalakshi, we are indeed grateful to our officers, first of all, Honorable Vice Chancellor, Professor P. Subramania Edipaditaya, Dr. Kishore Kumar C.K., Registrar, Mangalore University, and other officers for always supporting and encouraging the activities of IQAC. The Registrar, in fact, requested all chairpersons, coordinators to send all the life science students today to attend this lecture. So we are indeed grateful to him. Special thanks to Dr. Kedkar for coming to Mangalore University on his own initiative in the interest of research, in the interest of collaboration, and on this grand mission of planetary biodiversity mission. He is really a man on a mission. We wish him all the best for all his coming endeavors. Sir, as you are aware, Mangalore University, as Professor Krishna Kumar said, is located very strategically between our biodiverse Western Ghats on the one hand and our coastal biodiversity on the other hand. So we are very strategically located to be an important center for your mission. So please consider Mangalore University when you submit your proposal to Government of India. For your information, the state of Karnataka...
And faculty present.
For your information, the state of Karnataka, sir, occupies the majority part of Western Ghats. Okay, close to 23,000 square kilometers of Western Ghats are located in Karnataka. So Karnataka cannot be ignored when you go for such a big mission known as Planetary Biodiversity Mission of mapping all the biodiversity of the country. Karnataka cannot be ignored. Please remember. I think understanding the significance of this, Dr. Kedkar has embarked on his mission. Yesterday or day before, he was already in Bangalore University. Tomorrow he will be visiting 1 or 2 universities in Kerala. So thank you very much for coming to our university and for this excellent talk. Thank you. Thank you very much. On behalf of the university and IQAC, we extend a very small memento on behalf of our appreciation to you, sir. I request Professor Pattabi to please hand over a book to Dr. Kedkar. Dr. Kedkar, all of us gathered here are natural historians. We are all natural historians. This book is written by a true historian, none other than our late Professor Surendra Rao. So we request you to kindly read it. It is a book on history. We hope you enjoy it. It is a special gift from Mangalore University. Such a program is not possible without the active cooperation of all our life science departments. So it is really my privilege and honor to thank profusely Professor Krishna Kumar from the Department of Applied Botany for coming along with his colleagues and also bringing all the students of the Botany Department to this program. Thank you very much. We also thank Professor M.S. Mushtaq, Chairman, Department of Applied Zoology, for coming here with all of his colleagues, including research scholars and MSc students of zoology. Thank you very much. I wish to thank Chairperson of our own department, Professor Chandra M., for sending all the students, including all the faculty, to attend this program. Thank you. Professor Mushtaq, thank you for welcoming. Professor Krishna Kumar, thank you for introducing the speaker. I would like to thank our students, Sai Rupa, Veena Sharada and Vidya, our nightingales of Department of Biosciences, for the invocation. Thank you, Shreya, for very beautiful MC. Thanks to the faculty members of Biosciences Department for doing the registrations today. Thank you to Computer Center for facilitating online. I wish to inform Dr. Kedkar that our University College faculty and students listened to the online talk today. Last but not least, thanks to IQAC technical staff for all the assistance they have provided, for all the background work that they do to having made this program a successful one. Thank you one and all.
We request the audience to rise for the Mangalore University anthem.
ಸುತ್ತ ಕಣ್ಣ ಈ ವಿದ್ಯೆ ಹೊನ್ನ ಕಿರಣ ದಿನ ಪಾಠ ಪಠಣ ಗುರು ಬುದ್ಧಿ ವಚನ ಇದು ವಿದ್ಯಾದಾನ ತಾಣ